Okay, so this week's chapter or today's chapter are the cells. Let me put it in slideshow mode. Okay, let's put this up here. Okay, so cells, they are the structural unit of life or the repeating unit of life. I believe there was a not Zoom question, but a Kahoot Doody question based on that. It's the smallest unit of life. And we have about 500 to 100 trillion or 50 to 100 trillion cells in the body. A composite cell is a typical cell. And I'll show you a picture of what we're referring to here. You can usually see these, in online or in textbooks. They're usually like a planetary type model, I call them. They're round like that. And they have a nucleus in the center with a nucleolus and other organelles. But I will just tell you now that there are very few cells in the body that are actually round like that. I mean, some of them they're rather round, but they have very unusual shapes. And so when we're looking at these different parts of a cell and different organelles, depending on the cell's function, like whether it's an osteoclast, or whether it's a white blood cell, or whether it's a neuron, is its shapes can be very unique, given its function, but also the number of types of organelles, meaning perhaps some cells will have, very abundant mitochondria because like in muscle cells, they need to generate a lot of ATP to contract and relax and contract and relax. Osteoclasts that break down compact bone to release the calcium stored there so it can enter into the bloodstream. They are generally abundant in lysosomes. So although these are typical organelles, some cells have more or less of these organelles depending on, what they functionally do. But the major parts of the nucleus, the cytoplasm, and the cell membrane or plasma membrane. So here we have this model. We have a plasma membrane to the exterior that defines, what is the interior of the cell versus the exterior of the cell. We have different organelles, identified in different colors and different shapes. We have the nucleus and the nucleolus, the things, within the nucleus itself. So the cell nucleus, let me just move this thing up and away, is the control center of the cell. It's a genetic library with blueprints for nearly all your cellular proteins. So this is where our chromosomes are, which is where our DNA is. And when we covered nucleic acids in the chemistry family, we said DNA, RNA, are the genetic material of the cell. They are it's the recipe book and individual recipes. So all these different proteins, that are needed within the cell. So the chromosomes all stay in the nucleus, except when the nucleus or let's say the cell is going to replicate itself. And we're going to talk about that at the end of this chapter. But generally you want your chromosomes to stay compact, and bound in a membrane. And what we'll find is that most of our organelles in a cell have a similar phospholipid bilayer membrane surrounding it and keeping its contents nice and centrally located in this particular kind of a vesicle, so to speak, and it gives it or allows it to have some unique functions. Right, so the nuclear envelope is just another word for the nuclear plasma membrane. It's a double layered membrane, as you can see here in blue, that surrounds the nucleus. Right, so it separates the nucleoplasm, which is just the watery content within the nucleus itself, and the different organelles found there, such as RNA and chromosomes of DNA. And that membrane regulates the passage of material across it. So all membranes will do that. They regulate what enters and exits either the entire cell, if we're talking about the plasma membrane, or into and out of a particular organelle. The nucleolus is a dense collection of RNA and proteins, and it is the site of ribosome production. Ribosomes we're going to find either free-floating or attached to endoplasmic reticulum outside the nucleus, and they come from, or they're manufactured in the nucleolus. And then we have chromatin, which is just a fancy word for relaxed DNA and proteins in the nucleus. Right? This is where we store the information for the synthesis and the recipes for making our different proteins. You can see on the surface of the nuclear envelope, we have these little pores. And that's to either connect to rough endoplasmic reticulum, perhaps, or to just let certain materials that are of appropriate size, a size that can fit through the pore, enter into and out of the nucleus. Okay, so then we have the cell membrane, really referred to as the plasma membrane, and that controls what moves into and out of the cell in its entirety. It's selectively permeable and acts as an active barrier separating the intracellular fluid from the extracellular fluid. So it's a fatty layer surrounding the cell to just set up some kind of a wall and discriminating region between outside the cell versus inside the cell. And it's composed of a phospholipid bilayer. So, when we looked at chemistry in the lipid family, we said we had those phospholipids. The building blocks were one glycerol, two fatty acids, and a phosphate group. The phosphate group was polar covalently bonded, and the other building blocks were non-polar covalently bonded, like other triglycerides. And therefore, that created ends of the molecule that were polar covalently bonded and non-polar covalently bonded. And so the water-soluble polar covalent end, the hydrophilic end, we often refer to as the head, because it's represented as a ball. And then the fatty acid tails are the insoluble with water tails, because they're non-polar covalently bonded, the hydrophobic ends. And they arrange themselves in such a way that the fatty ends point toward each other and the phosphate groups point out to the external and internal environment. And thus that fat, because it has a tendency to repel either large molecules that are polar covalently bonded, or most polar covalently bonded or charged entities, repels them because they have incompatible bond types. So that's what we mean by being, semi-permeable. Things that are similarly bonded as fats that are primarily non-polar covalently bonded or neutral can readily kind of enter and exit the cell because they have no problem with passing between the fatty acid tails of a phospholipid. We also have proteins there that act as receptors, pores, channels, carriers, enzymes, self-cell markers. We said that in the protein family, that's where we have molecules that perform the most complicated and sophisticated functions in the cell, and they're all going to be proteins. And always remember, proteins, they are the only family that has that primary, secondary, tertiary, and quaternary structure. And it's the hydrogen bonds in the secondary, tertiary, and quaternary structures that keep them in the shape that they need to be in to perform their particular functions. And that those hydrogen bonds can be broken during denaturation. What causes denaturation? Environments of high temperature or high body temperature and low pH. Technically extremes of pH, but in the body we generally just have low pH with rare exceptions. Okay, so transportation across the membrane, it's selectively permeable, which means that it can also be used to create and maintain different concentration gradients of different molecules, outside versus inside. What we're going to learn is that, the cation that's in highest concentration inside the cell will be potassium. The one in highest concentration outside the cell is going to be sodium. And we have what's called the sodium potassium pump that keeps it that way. Right, so transportation across the membrane, we have ion channels. These are pores which ions can pass through. So a channel is like a pore. It has a very narrow diameter, so small things can pass through it. It connects the external environment, the extracellular fluid to the intracellular fluid, the outside of the cell to the inside of the cell. So this channel lets small things with charge, pass right through it. Then we have transport or carrier molecules that selectively move polar molecules across the membrane. So these molecules also have charge, but the transporting molecule or carrier molecule is generally attaches to and transports larger molecules than just these little ions. So glucose and amino acids are good examples of that. And then we have receptor, that recognize and bond to specific molecules. Let's say they're capable of binding to the hormone insulin, which lets the cell know that we have elevated blood glucose levels. And now it might start to want to actively transport some of that glucose that's outside of the cell into the cell so we can use it to burn, in our mitochondria, so to speak, and form ATP, form energy. Okay, so then we have the cytoplasm. So we talked about the nucleus, we talked about the plasma membrane, and then we have the cytoplasm. And the cytoplasm includes the cytosol, which is the watery environment, and all the organelles found in it. I will say that we're all kind of can be faulted with using cytoplasm and cytosol interchangeably, but technically, the cytoplasm is everything between the nuclear membrane and the plasma membrane. And that would include the watery environment that all these chemical reactions occur in, and all the organelles that are suspended in that fluid. So examples of fluid are the endoplasmic reticulums. We have two types. We have rough and smooth. So an endoplasmic reticulum are these connected sacs and canals with one another. So if we look at this one, we have two here. We have in red with dots on it, the rough endoplasmic reticulum, and then off to the right, we have a smooth endoplasmic reticulum. And the rough kind of gives you an idea, even though they've cut it, so you could look down and see that these sacs kind of are interwoven with one another, is that you have to picture that expanding much more to the right and left and superiorly, that these big areas of interconnected sacs. And they transport molecules through there. The rough endoplasmic reticulum will be integral in forming phospholipids and different proteins. And the smooth endoplasmic reticulum, primarily works on lipid synthesis and breaking down of drugs and detoxification. But here we have the first is the rough endoplasmic reticulum. It's called rough because ribosomes stud the surface and it gives it a rough appearance. And this is where we synthesize membranal integral proteins and phospholipids, in there. The smooth endoplasmic reticulum is really associated with lipid synthesis, especially what's called chylomicrons. They become added to proteins from the rough endoplasmic reticulum. They also are active in breaking down different drugs and cells in your liver. Your liver detoxifies a lot of the, especially pharmaceutical type drugs, but alcohol and different substances in the body. So those cells, are going to have a lot of smooth endoplasmic reticulum. This is where peroxisomes, are formed. And I used to have on here, and I think it's later in the lysosomes. Lysosomes pretty much bud off the rough ER or the Golgi apparatus, and the peroxisomes, we're going to talk about those, bud off of the smooth endoplasmic reticulum. Ribosomes are free floating or connected to the endoplasmic reticulum or to the rough endoplasmic reticulum. So they may be free floating. Sometimes you'll see them in the cytosol, these little black dots there. Those are some ribosomes. And they function in amino acid assembly and thus protein synthesis. So when we form proteins, we make the proteins, we said we start with a primary sequence and secondary, tertiary, and quaternary. And the primary sequence is made by these ribosomes. There will be a form of RNA that is essentially carrying the recipe of the sequence of maybe the 50, 100, 150, 175 amino acids that need to be in a particular sequence in that initial sequence. And the ribosomes, are reading the message from the RNA, and then one of the types of RNA, like transfer RNA, is going to bring each amino acid to the ribosome, and it's going to link them together. So you have this string of amino acids, and it'll frequently enter into the rough endoplasmic reticulum because they're frequently on the surface, of that endoplasmic reticulum, and it'll be worked on in the RER, and then it buds off generally and gets transported to the Golgi apparatus. Some of the products of the RER is going to go right to the smooth endoplasmic reticulum. But frequently the proteins that are going to be replacing maybe other receptors on the plasma membrane will be modified in the Golgi apparatus. So the ribosomes are part one of multiple organelles that are associated with protein synthesis. Other organelles include the mitochondria. right? So it's often referred to as the powerhouse of the cell, when you were in high school or elementary school. But we really want to kind of up the ante there on our description of it. And this is the organelle that produces the ATP, which is the energy currency of the cell, through aerobic cellular respiration. And so what it means is it's going to be aerobic, it's going to require oxygen, and it's going to go through 4 different phases of breaking down, preferably one glucose molecule, into netting about 30 to 34 ATP. And it does so, it begins that process outside the mitochondria, and then the oxygen requiring steps will be within the mitochondria. So for a cell that does a lot of physical work, like muscle tissue, it's going to have an abundance of mitochondria. Other organelles include vesicles. Vesicles, are it's kind of like anemia. It's a general term. It's a sac that contains or stores something in the center. And when we know really what kind of material is in the center, then we might call it a peroxisome or a lysosome. If it's a sac, that was created when a white blood cell engulfed some bacteria, then we're going to call it a phagosome. But a vesicle is just generally, a balloon that's filled with something, maybe with air or a water balloon or, jello inside or whatever. It's just a sac. And we often grab those sacs and pull them around the cell and transport them to where they're needed. The Golgi apparatus is another group of stacks of sacs that are generally in our models are colored brown. They're not as numerous and widespread as other organelles, but we have the Golgi apparatus. And as material passes from one sac to another, it becomes further modified and concentrated and specialized to ultimately produce the receptor or, protein product we're trying to make. Right, so this picture is good. We're not going to look at an extraordinary detail of protein synthesis. We are going to look at where protein synthesis starts and what kind of specific, functions or semi-specific functions are performed there and what we call that, because we do that in the lab here. But this just helps you kind of see the relationship of the rough ER to the Golgi apparatus and how the material will pass through these sacs and then they bud off. And once it buds off, it takes a part of the membrane of that rough endoplasmic reticulum and forms a vesicle. So, over to the middle top area, you can see those little blue balls floating around. Well, that generically is called a vesicle. Now, some of those might be dragged over to the smooth endoplasmic reticulum and acted on, but many of them will be passed to the Golgi apparatus here in green, where we have some more sacs all interconnected to one another, and it will pass through layer to layer to layer until it's the production of it is finalized, and then it buds off there and forms those vesicles. So depending on what's inside the vesicle, it will determine whether it just maybe merges with the plasma membrane, as it's showing you here. And the substance, those little purple balls in the center, are just being what's called exocytotically released. That vessel will, or the vesicle will merge with the plasma membrane and incorporate itself and then spit out its contents, essentially. Sometimes, the vesicle wall contains receptors that need to incorporate themselves into the plasma membrane and maybe replace some of those protein receptors or pumps that have now kind of broken down, because the plasma membrane of these organelles won't last forever. The cells live for a pretty long period of time, and they're able to replace worn out organelles and worn out surfaces and areas of its plasma membrane. And this is a method in which it does that. So you can see how these vesicles kind of bud off and go from one to another, and then they are released in vesicles in the cytosol, and we have infrastructure in there that often grabs those and pulls them around the cell to the area in the cell that requires them. Okay, so when we look at protein synthesis, and I ask you questions in lecture as well as you will be asked questions of this in lab, and it generally will use models to represent, DNA to DNA, which is called, or show you two DNA molecules. If I start with DNA and I end with more DNA, it's called DNA replication. And that occurs in the nucleus. So we have a few steps or processes of protein synthesis that begin in the nucleus and then end up with, a vesicle. So whenever we have protein synthesis, we need to be capable first of replicating our DNA. All right, so we need to be able to have our DNA and make a copy of DNA. And that's called DNA replication. It occurs in the nucleus. Then we need to be able or capable of making a copy of the DNA that's capable of leaving the nucleus, because the DNA and the chromosomes are going to stay in the nucleus until we have to make a copy of that cell. So transcription is when we start with DNA and we end up with RNA, which is a copy of the DNA that can leave the nucleus and now go out to the ribosomes. So we've made a copy of the recipe for some proteins. Now that can leave the nucleus and go out into the cytosol, and that's where the ribosomes come in. So the ribosomes translate that message. In the nucleus, we write it down in the form of RNA, which is transcription. Now we take that recipe out to the ribosomes, and now they read it or they translate it into an amino acid sequence. Then it passes through the RER and ultimately to the Golgi body, where they're modified. So the Golgi body is associated with modification of proteins. And then it buds off, all right, to form different vesicles. And generally those vesicles are going to be incorporated in the plasma membrane, and we will exocytotically release the contents. Right? So the processes are DNA replication, transcription, translation, modification of proteins, and exocytosis. And then these are the organelles that are responsible for performing that function. So moving on to other organelles, we have lysosomes. And lysosomes are generally, I call them the blue jelly beans. There's often a blue jelly bean in the cytosol of the cell models that we look at in lab, and even in the lab book, that in case you're doing this online, the lab book. And they contain digestive enzymes. So their enzymes are designed to break down more physical components of something. They, may merge with worn out organelles in the cell and merge with it, and then secrete these enzymes onto it so it breaks down, and then that vesicle, that lysosome, will merge with the plasma membrane and release all that waste, to the extracellular fluid. If it happens to be a white blood cell that engulfs bacteria, then once that is engulfed endocytotically, then this lysosome merges with it and it releases these enzymes on the surface of that bacteria, a virus, and it breaks it down. And then again, it will often be transported to the plasma membrane, where it can incorporate itself and kind of eject the contents of that lysosome and its degraded bacteria out into the extracellular fluid. So they generally digest with enzymes, larger substances, molecules, organelles, bacteria, viruses. Now they can contain, especially in certain cells, like an osteoclast. Osteoclasts are those bone cells that break down bone. So they are going to have more lysosomes than the average bear, that contains the digestive enzymes to break down compact bone. And we do that because we need to take some of the calcium that's stored there and put it into the bloodstream so that we can elevate our blood calcium levels. Okay, then we have centrosomes or /centrioles. They're often used interchangeably. These are organelles that will replicate when we're ready to try to replicate the cell. So these centrioles will replicate, and they travel to opposite ends of the cell. We're going to look at that, toward the end or at the end of this chapter, really. And they're capable of making what's called mitotic spindles. And they're essentially, it's like an erector set, this, this framework that will be built upon and be extended so that they create these kind of finger-like projections to grab onto the chromosomes and pull them apart so that one copy goes to one cell and one copy goes to another. Right? So they're active in distributing chromosomes during cellular division. Then we have peroxisomes. Now, peroxisomes have digestive enzymes, but for sort of a different purpose. Peroxisomes, and I will say on the cell models down in lab, those models don't have an example of a peroxisome. They do have a lysosome, but they don't have an example of a peroxisome. But they are similar in their appearance as a lysosome or any other vesicle. They're a membrane sac that contains something in the center. But these enzymes, are active in detoxifying toxic substances such as free radicals, and break down organic molecules. So they're enzyme-containing sacs that originate and bud off from the smooth endoplasmic reticulum. Unlike the lysosomes, they're budding off the Golgi apparatus, and arguably the rough endoplasmic reticulum, depends, but it definitely won't bud off of a smooth endoplasmic reticulum. That's where the peroxisomes come from. And they have two major detoxifiers. They have oxidases and catalases. And oxidases are used to convert hydrogen peroxide, or used to convert toxins really to hydrogen peroxide. And because hydrogen peroxide is really a toxin in the cell, we need to further neutralize it. So then we use what's called a catalase to act upon it, and then it'll convert it to water. If you've taken any microbiology, at HCC, you can be an AMP1 and take microbiology. Here you had to have had and completed as a prerequisite AMP1 and AMP2 in microbiology. So sometimes, I have students that are familiar with oxidase tests and catalase tests that you use in microbiology, and you're going to use this there. It will come back because, once you are trying to identify an organism, one of the first things you do is determine whether it's gram-positive or gram-negative. And then if it's gram-positive, you perform a series of tests, and one of them is going to be this catalase test. Okay, so that takes us to our first Kahoot. If you'd like to play, let me stop my share and re-share my share. Reshare my share. Just give that a moment. Hold on. There it is. Yay. Okay, let's get started. Nice. Good. The presence of ribosomes on its surface. Good. 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 Centrioles. Good. So, something I just want to give you like a hint on. If we're talking about really digestive enzymes, quote unquote, then we're really talking about lysosomes, and kind of neutralizing, detoxifying, enzymes are really the job of the peroxisomes. Good. All right, nicely done. These are, hold on, let me just get out of this. That material makes really good, structure function kinds of questions. If people often ask, how do you approach studying this? I think flashcards are handy, but and it serves a purpose to rewrite notes to a certain extent. I don't know if I buy a case of index cards for that purpose, but you can do something like this with a PowerPoint and put it in slideshow mode and essentially create, flashcards, and you have an organelle, and then the next one, let's say paging down, would be the function of that organelle. So those are really good ones to use as quick flashcards because you don't want to miss those easy questions. Okay, so now we're going to start talking about more of the physiology within the cells, of which there's a good amount. And I will say that, at least these basic terms, of the difference between like hydrostatic and osmotic pressures, osmosis versus diffusion, the sodium potassium pump, these things, they come up, over and over and over again. So they're definitely, your foundation. All right, so when it comes to movements into and out of the cell, we basically have two basic categories. We either have passive processes or active processes, and passive processes require no energy whatsoever, and active processes require energy in ATP. So those that no energy are required are simple diffusion, facilitated diffusion, osmosis, and filtration. Essentially, and I write down there below, anything with diffusion or osmosis in the title, it falls into the passive category, because I might just ask you which the following is not a passive process, or which of the following is an active, movement into and out of the cell or a process. Filtration, I generally don't ask that, but filtration will occur. You are like making coffee is an example of how we filter something. We have to pump the water, or put the water above the filter that might contain, let's say, coffee in this case, and as the water passes over the grounds, gravity pulls on it, and it takes whatever molecules it can interact with, and it passes through the filter and down into your coffee pot or into your cup. That's filtration. It just separates things by size. Large size, the large grounds, stay do not pass through the filter generally, and that's why you have to empty it after you make coffee. So a filter separates things by size. Active processes require energy. Things like active transport, endocytosis, exocytosis, and transcytosis. So we're going to talk about these, diffusion versus osmosis, especially, right? Diffusion is the movement of a substance or molecule from an area of high concentration to an area of low concentration. Essentially, you're letting it just roll downhill. Just this in the same, that's not the same thing, but we were talking about elements and atoms being driven to fill its outer valence shell, which is the basis of the chemical bonds that are formed in nature. And that happens. You don't have to push it. You don't have to encourage it. It just happens on its own. And diffusion and osmosis, given a pathway for them to follow, happens on its own as well. Everything wants to be equal, equally spread out. And so, that is this driving force. The nonpolar hydrophobic lipid core of the plasma membrane blocks the diffusion of most molecules. Those fatty acid tails form a fatty layer around the cell. The molecules that are able to passively diffuse to the membrane include lipid-soluble molecules, the molecules that have a similar bond type as the fatty acid tails. Nonpolar substances, because they're not going to be repelled, they're going to be neutral. A lot of oxygen, carbon dioxide gases, steroids, because they're in the fat family, fat-soluble vitamins. That's why we would say that they're fat-soluble, because they are non-polar covalently bonded, or they're neutral, A, D, E, and K, in small amounts of water, urea, and alcohols. So water, when you have a pretty good concentration gradient between inside the cell and outside the cell, as far as the water concentration, those water molecules can either squeak through the fatty acid tails, or there will be some little pores on the surface called aquaporins, and that allows the passage of water or water molecules to pass through it. Maybe not a lot all at once, but it'll let them kind of squeak through. The rate of movement can change. The rate of diffusion increases when the concentration gradient increases. When temperature increases to a certain point, you don't want it to be too high where it cooks all your proteins, but the more you increase temperature, you increase molecular movement, especially if a substance was frozen and now it's at room temperature. And when we increase the surface area for transport to occur, we decrease the rate of movement, with some other examples. Well, first of all, if we decrease the concentration gradient, if we decrease the temperature, if we decrease the surface area, clearly, but we also decrease the rate as the molecular size increases. If the diffusional distance increases, if the distance, the thickness of the membrane, or the thickness of a region, becomes much thicker or wider, it's harder for the molecules to diffuse through them. And larger molecules are generally harder to move anyway. The diffusional distance and pathway becomes very important when we talk about the respiratory system, because when it comes to what's called the respiratory membrane, it's where the smallest microscopic working gas exchange area of your lung is. It's composed of one layer of simple squamous epithelium, meets a capillary, which is also one layer of simple squamous epithelium. So two very thin paper-like structures coming together. And because they're very thin and they have kind of pores between each cell, or it's not really a pore, but they have what's called fenestrations. These cells don't abut and form very tight junctions at all. And when you have them very, very close together, oxygen in your lungs can diffuse into your blood because it's low in your blood. And the carbon dioxide that it's at high level in your blood, in your lungs, can go from the blood into the lung tissue, or into the alveoli, I should say, the working unit of your lung, and then you can exhale it out. And that the thickness of that respiratory membrane, where this gas exchange, must occur within like a quarter of a second, a fraction of a second. You need to have those membranes very thin and approximated to one another to allow diffusion to occur very, very rapidly. If it gets thicker, with, let's say, pulmonary edema or different, growths in the tissue and scar tissue, if it's thicker, and denser, you're not going to have the same ability to diffuse. So carbon dioxide has a tendency to build up in the blood, and oxygen becomes, lower in its content. All right, then we have facilitated diffusion. So all that means is that you're going to have a carrier protein or a channel protein of some kind to allow the molecule to go from one side to another. It's generally for larger molecules. They can't just squeak through maybe an aquapore or a little pore that would allow ion. This is generally sugar molecules, or amino acids that can either go from outside the cell to inside the cell or inside the cell to outside the cell. But it needs sort of a bridge or a tunnel to use, and that's what these molecules will do. So these are these purple molecules embedded in the plasma membrane. I just want to stay here for a second. And this allows us to actually get a very good idea of what the plasma membrane and the phospholipid bilayer looks like. Do you see the little ball with the two fatty acid tails? You have two layers. That's the bilayer of those phospholipids, a ball and two tails. And you see how those tails orient themselves because they have similar bond types. They're all nonpolar covalently bonded. And the phosphate groups that are polar and hydrophilic point to the outside of the cell and the inside of the cell. And they are maintained there often by some cholesterols and steroids that are embedded between, the fatty acid tails. And because of bond like dissolves in like, similarly bonded molecules like to be in similarly charged somewhat solvents. So polar covalently molecules or ions that have charge like to be in water because it's polar covalently bonded, and fats like to be in other non-polar covalently bonded solvents, is that they'll maintain their orientation there. And that is actually the fat layer that essentially forms a plasma membrane, or the border between the outside of the cell and the inside of the cell. Okay, osmosis, H2O, that's how you're going to keep that straight, because osmosis is generally, for all intents and purposes, referring to the movement of water across a semi-permeable membrane. So the movement of solvents, such as water, across a selectively permeable membrane from a region of high concentration to low concentration. All right, so water is going to do the same thing. If you have a high concentration of water outside the cell compared to inside the cell, that's called being in a hypotonic solution, then the water molecules are the force or the draw is the water to enter into the cell so that we have equal amount of water on both sides of that plasma membrane. I mean, it's just a passive driving force. So water diffuses through plasma membranes, or it can. It can pass through the lipid bilayer. Even though water is polar, it is so small that some molecules can sneak past those nonpolar phospholipid tails. Or if there are aquaporins, which are specific little channels, for water, then they will pass through those. We see a lot of the aquaporins when we look at some of the types of cells and how we're going to reabsorb water in the urinary system. Osmosis and osmotic pressure. So the movement of water causes pressures, all right? Hydrostatic pressure of water inside of the cell or vessel is the push outward. And I'm going to give you an example of that in a minute. We especially talk about this generally. It's the hydrostatic pressure is often going to be the push outward that we refer to in most of the systems we're looking at. Not that it can't work in reverse. All right, so let's use the example of a hose, on the outside of your house. And you turn the water on and the water passes through the hose. And let's say you have a nozzle at the end of the hose. And until you squeeze the nozzle, the water stays in the hose, right? The water does not permeate through the walls of the hose because you have layers of rubber and things that keep it in there. But the water pump, inside the house is pumping water, into the hose. So then what you can do is squeeze the handle. Right? And what happens? The water comes shooting out. It becomes shooting out because the pressure inside the hose exceeds the pressure outside. And so the water follows that pathway. Right? It's a push outward generally, push out. The osmotic pressure, is generally different. The osmotic pressure is drawing or pulling water toward you. Right? It's a tendency of water to move into a cell or vessel by what's called osmosis. More solutes inside the cell, the higher the osmotic pressure. All right, so let me give you an example of this that you can hopefully relate to. And it's kind of hard to do that because you don't know how capillaries work. But when we transport blood, we transport them in arteries, ultimately, to veins. Arteries transport blood away from the heart, and veins transport blood to the heart. But between the arterial side and the venous side, all the blood vessels narrow down to these tiny, tiny little vessels called capillaries. And they are of a diameter that allows one red blood cell at a time to pass through it. And prior to that, in the arteries and the arterioles, we have multiple layers of the vessel wall, kind of like a hose, that doesn't allow material really inside that tube from the blood to leave the cardiovascular, so leave those blood vessels and go out into the interstitial spaces. But when it finally travels in and passes through a capillary, all of a sudden we have this very narrow diameter hose, and it's only composed of one layer of that simple squamous epithelium, a layer of flattened cells, that essentially have little gaps between them. And now as the blood enters that capillary, molecules are no longer contained within the vessel wall. They can actually fit through these little open windows and leave the blood and go into what's called the interstitial spaces. And that's because we're pumping it so hard from the left ventricle. Prior to that, you didn't have an open window. Now you get to a capillary, now there's some open windows. And so it's just like squeezing the nozzle on the hose. You have a pathway to go, you have a place to go. And so anything that can fit through the window is going to leave. But as that blood travels from the arterial side to the venous side, because it drops off the oxygen and now carbon dioxide diffuses into the blood because it's high concentration outside these blood vessels and low concentration inside, is that as the blood passes across that capillary, the blood inside that capillary, now has a very high osmotic pressure in it because it's very concentrated with substances and less with water. And so now it has a tendency to pull some of that fluid that we just lost entering the capillary. Now we draw it kind of back into the capillary. And then it transported in what's called venules and veins and back to the right side of the heart. So when we're looking at these pressures generally between hydrostatic and osmotic pressure, the hydrostatic pressure is often this push outward, out of a blood vessel, that occurs in these repeating units of the kidneys, called nephrons, because the blood gets pumped in with such force that we can filter it, and molecules that can get through these little open windows will get through and leave the bloodstream and now start passing down another pathway. That's hydrostatic pressure, a push out. Osmotic pressure is pulling it back in, is pulling water back in. So osmosis, a living cell has limits to how much water can enter it. Each cell, cells can grow, but they can't grow to be the size of a football. The nucleus and all the organelles in there can really only maintain a certain amount of plasma membrane and take care of everything in there. It's like when you only make, when you make a certain amount of money, you can really only afford to maybe maintain a certain size house really well, or car or something. If it gets too big and you have a mansion and you're only making 50 grand a year or 30 grand a year, there's no way you're going to keep up with all the maintenance, of a mansion, a 5,000 square foot home, and keep the yard and keep everything. So kind of think of it that way. So the cell can't have an abundance of water inside the cell. If it's too concentrated inside and the osmolarity, or the osmotic gradient is too high in the cell versus outside of the cell, it's going to draw fluid into the cell to the point that it just expands and breaks. So we're going to look at the different the tonicities of different fluids that body cells often reside in. So tonicity is the ability of a solution to change the shape or tone of cells by altering the cell's internal water volume. If the ratio of solute to solvent, is dramatically different outside the cell versus inside the cell, and we're going to consider that most of them, we're going to assume that the inside of the cell isn't dramatically changing on its own. Is that the fluid environment a cell sits in, like a red blood cell sitting in plasma of blood, what effect does that have on your red blood cells, and ultimately out in the body as well? It's like, well, what will, the effect of the osmolality, of interstitial fluids have on cells that's in environment? Will it cause it to shrink or expand, or will it have no particular effect at all? So an isotonic solution means that you have the same osmolarity inside the cell versus outside the cell. It means that, for instance, let's say inside the cell, we have 75% water and 25% everything else solute. Anything else is suspended in the watery environment. 75% water and 25% solute of some kind. And the solution it's sitting in the watery environment is also 75% and 25% solute. It means that osmolarity inside the cell is the same as outside the cell. Inside the cell, we're 75% water. Outside the cell, we're 75% water. So there's no concentration gradient, or there is not a concentration gradient of water between inside the cell and outside the cell. They're the same. So really, there's no net movement of water. It kind of flows a little bit into and out of the cell, but it makes really no effect on it whatsoever. But a hypertonic solution a cell might sit in means that it has a higher osmolarity, right, outside the cell versus inside the cell. Or well, let me give you an example of that. We said that the first example, the cell was 75% water inside and solute was 25%. Outside the cell, let's say that now the solution it's sitting in is 10% water and 90% solute. So it's really kind of thick, very salty, kind of like salt water, like out in the bay. Water is higher concentration inside the cell at 75% and only 10% outside the cell. So if osmosis is the movement of water from an area of high concentration to low, it's going to go from the interior of the cell to the extracellular fluid area, which means it's going to kind of suck the water out of that cell. It's going to cause it to shrink. And thus that's called crenation. Or the opposite could happen. We could have a cell in a hypotonic solution, so let me give you an example of that. This is where you have a lower osmolarity inside the cell, so that water flows into the cell, resulting in tissues, or the cell swelling. So let's take that same example. We have a cell that is 75% water inside and 25% solute inside the cell. A hypotonic solution, now it's going to be the opposite of what the hypertonic was. We're going to be maybe 90% water and 10% substance that we're transporting in their solute. So the watery environment is 90% water, and the inside the cell is only 75% water. It's mostly water outside versus inside. So what direction will the water move from where it's in high concentration to low? Where it's in higher concentration outside the cell, and it's going to be drawn into the cell and cause it to expand and possibly lyse. So when you think about hypertonic and hypotonic solutions, we talk about cells like this. If we're saying the solution is hypertonic, it means it's very thick, very little water compared to the inside of the cell. So if the solution is hypertonic, then the interior of the cell is comparatively the hypotonic side. And if a cell is in a hypotonic solution, means the solution has a much higher amount or concentration of water in the environment, and less inside the cell. If the solution is hypotonic, then the interior of the cell is relatively hypertonic. Right? Does that make sense? So think of it this way, wherever the hypertonic side is, the water is going to be attracted to it. Where salt goes and lots of solutes go, water follows. So where salt goes, water goes. So if the solution it's sitting in is hypertonic, the water gets drawn to the hypertonic region from inside the cell to out, and it causes it to shrink. If the solution a cell is sitting in is hypotonic, then the interior of the cell is relatively hyper. Now we can predict the direction. Water will go from outside the cell to inside the cell and cause it to expand or lyse. This shows you how, or some examples of the response of red blood cells in these different solutions. If we're in an isotonic solution, which is A, is that there's no real difference between the concentration or the osmolarity inside the cell versus outside the cell. The water concentrations are pretty much the same. And this is assuming that the solute is not going to move. We're just looking at the movement of water. Then we have a hypertonic solution. That's essentially we're hypertonic outside the cell and hypotonic comparatively inside the cell. So it draws water out of the cell and causes it to shrink. A hypotonic solution has the opposite. You have essentially the interior of the cell hypertonic compared to the outside of the cell, which is hypotonic. And so now water is in greater concentration outside the cell. It wants to be equal on both sides of this plasma membrane. And so it enters the cell and it causes it to expand. This becomes the basis of how the different types of intravenous IV fluids that can be administered to a patient. Because in this example, where we're just talking about, and this is initially the worst effect, is, well, what's happening to my red blood cells? Because that's really important. You need to be able to be transporting oxygen, and within, the plasma, most of your carbon dioxide and things. So, the healthy nature of your red blood cells is really, really important for survival. But as things progress, or particular pathologies exist, and it might not necessarily be pathology, but it's some form of abnormal allocation of fluids within the body, is that you can use different intravenous solutions. So we're talking about a solution that we're going to put into the bloodstream to help affect or change where fluids are located in the body. Not something you're going to drink necessarily, but you're going to administer into the cardiovascular system. So intravenous solutions of different tonicities can be given to patients suffering different ailments, or issues. Isotonic solutions are commonly given when blood volume needs to be increased quickly. So an example of that would be severe dehydration. If the number and quality of your red blood cells and white blood cells and platelets are good, you're just really, really dehydrated, you just set them up with an IV solution, of saline pretty much. And, it's an isotonic, it's the same osmolality as that which should normally be in blood, and you're just going to increase the amount of water and restore what's called the plasma portion, which should be 55% of your blood, and that can be done, rather quickly. But there are examples of using hypertonic solutions and hypotonic solutions. Hypertonic solutions are often given to people who have edema systemically in the body, not just your ankle swollen because you sprain your ankle and you have inflammation occurring there and you have injury. That's not that you don't want to rest, ice, compress, and elevate type of a thing, but it's not something that requires an IV application. It's when it's generally systemic, throughout the whole body. And so when you have fluid that is not being drawn back into the bloodstream or what's called the lymphatic system, that's what swelling in the hands, the feet, in the abdomen, perhaps because you have maybe problems with, maybe with your kidneys, or you have congestive heart failure, that you have fluid accumulating, in the tissues, and that's getting drawn back in, let's say. Although those can be contraindications to using the solution, because once you put the hypertonic solution in the blood, it's going to draw fluid to it. And you have to administer it very carefully, because if you do have a weakened heart or you do have some kind of renal disease, you don't want to overcome that organ's ability to work. So, you'd have to do it very cautiously with patients, like that. Hypotonic solutions may be given in cases of, you don't know what this is, but you can hold on to it to when you get into either AMP2 or into nursing school, is ketoacidosis or hyperglycemia. Hyperglycemia means higher than normal amount of glucose in blood. And whenever we have more molecules like that, in the blood, it changes, the thickness sort of blood. And so it would be a good idea, to have a hypotonic solution administered to kind of, what can I say, dilute it so that your blood doesn't get too thick. So where blood plasma is rather hypertonic, has less fluid and water in it than we want, and it's creating the cells, the red blood cells, because that can result in dangerous lysing of red blood cells and white blood cells. Okay, so there are certain circumstances, where you can use a hypertonic solution versus a hypotonic solution, and that's to bring things back to homeostasis. Okay, active transport. So we looked at passive transports. We need to look at active. The carrier molecules can transport substances across a membrane from regions of low concentration to high concentration, or uphill, and it requires ATP. All right, so before, diffusion and osmosis were passive and didn't require energy because they were really moving downhill, essentially rolling downhill. They were going from an area of high concentration to low concentration, until everything hopefully gets equal, on both sides. That's this driving, force. If so, active transport means we're working in the opposite direction. We're pushing things uphill. We're trying to keep, more sodium outside the cell versus inside the cell. And that requires ATP, that requires energy. And an example of that is right now, it got pretty darn hot today, it's in the 90s, probably lower 90s, and in your house, you generally don't want it to be 90 degrees, right? So what do? You pump cold air into your house or into your apartment or into this building that I'm sitting in. Right? Now, if I stopped pumping cold air in here, and I just stopped, and if these were perfectly insulated walls, which they're not, but let's say they were, it would stay cold in here. It would stay, whatever this is, 76, 78 degrees inside, and let's say 88 degrees outside. And if I opened my windows, what's going to happen? Well, the cold air from inside goes out the window, right? And the hot air from outside comes inside. That's passive movement. That's that is diffusion, right? I don't have to push it. Once I open the window and I have a pathway, and the interior of this room is eventually going to become the same temperature as outside. All right, that happens without any energy put in. But if I want to get this room back cooler again and keep hot air out and cold air in, I need to pump more cold air in here, which means the compressor has to work. And I think most of us know, to get your compressor to work and pump cold air, is it free? No, it is not. You need to have electricity into this system. So it costs something to pump all this stuff, to maintain hot, warm outside and cooler inside. Right. So that is an example, or those are active transport mechanisms. Right. They generally will require a carrier protein. And the solutes move against the concentration gradients from an area of low to high. Right? If it's going from high to low, it doesn't need any energy. It just diffuses. It's totally passive. So these things must be moving from an area of low concentration to high concentration. The sodium potassium pump is the pump we see most frequently for this. Right? And this is literally, a pump. I mean, other active transport mechanisms, we're going to talk about that, endocytosis and exocytosis, but the sodium potassium pump is designed to keep sodium outside the cell and potassium inside the cell. Basically is an enzyme called the sodium potassium ATPase that pumps 3 sodium out of the cell and 2 potassium back into the cell. Sodium is the cation highest concentration outside the cell, and potassium is the cation highest concentration inside your body cells forever and ever and ever. okay? Because where sodium goes, chloride goes, and where salt goes, water goes. So if there's too much sodium and ultimately chloride in the cell, it's going to, again, osmotically or draw that water from outside the cell inside the cell. The osmolarity in the cell is going to become too high. It's going to act like a hypertonic solution and draw water toward it. All right, so it's if a cell can control where sodium is located, in doing so, it's controlling where water is located. And that way it can maintain its nice little cell size so it doesn't get too big or too small. If it needs to get, if it's creating too much, it may, allow some of that sodium to reside inside the cell and let it draw some of the water into it and let it re-expand and get back to its normal size. But that sodium potassium pump pumps sodium in a particular direction from inside the cell out, 3 of them, and 2 potassium from outside the cell back in. Now, this distribution of ions is imperative not only in all your general, body cells, but in excitable cells like nerves and muscle cells. Okay, vesicular transport, we said that is active process also. This is when we're going to be moving and dragging around maybe these different vesicles, that contain different substances. So it involves transport of large particles, macromolecules, fluids, maybe bacteria that you just engulfed in membranous sacs called vesicles, and it requires cellular energy. So we have 2 basic types. We're either taking something into the cell or we're releasing something, to the exterior of the cell. So endo means within, cytosis or cell, and exo means outside or without the cell. So endocytosis is when the cell engulfs something or takes in a substance into the cell, forming a vesicle. We have 3 types. We have pinocytosis, phagocytosis, and receptor-mediated endocytosis. So we're really referring to the difference between pinocytosis and phagocytosis is what you're taking in. If you're pretty much just taking in water, then it's called pinocytosis or cell drinking. If you're taking in some kind of a solid, especially something like a white blood cell that might be engulfing a virus or a bacteria, it's called phagocytosis. And receptor-mediated endocytosis requires the binding of that substance to a membrane on the outer surface of the cell. So it goes, ooh, now that molecule, it recognizes the molecule is outside the cell, and it goes, ooh, I want to take in that one, And so sometimes it's a byproduct of insulin binding to a receptor that's saying that there's glucose outside the cell, and now it's time to start to engulf that or either allow it to be transported across a membranous protein, a transport protein. Okay, so all that means receptor mediated is that you have to have the substance bind to the surface first, and then that signals the cell that it might want to engulf it and take it in. So looking at the images here, you have a cell, right? Here's this plasma membrane. Something on the surface of that cell needs to be, or it's desired to take it into the interior of the cell. So it pinches inward. It like invaginates, so to speak. And then the sides of the plasma membrane meet and close off, and now it buds off into a vesicle inside the cell. That's endocytosis, taking things into the cell. Right? Exocytosis, see how these diagrams went to left to right, to the left, to the substance, something binds to maybe might be receptor mediated, on the surface of that membrane. It signals it to pinch inward and create a vesicle. Phagocytosis will go from right to left. It will be a substance that's in a vesicle, right? And then it merges with the membrane, incorporates within it, and then spits out its contents. Like that other picture that we were looking at with protein synthesis. So it's just the opposite. So it's the reverse of endocytosis. You're going to have a vesicle within the cell that needs to merge with the membrane and incorporates itself, and then it spits the contents outside. Substance in a vesicle fuse with the cell membrane, and the contents are released to the outside of the cell. But all active processes requiring ATP. Okay, so that takes us to another Kahoot Doody for those who want to play. Let me stop that other share and let that kick in. Look at all these people. Come on, you guys, why don't you play? You can always use somebody else's name. It's okay. There's anonymity. Let's hope that this gets going. There we go. Continue and sign it. Can everybody see it? Yep, I guess you can. Good. All right. If you're still logging in, the password will be down in the lower left-hand corner in case I start this a little prematurely, but you can still join in. So good. That's the osmotic push to pull things in. Nice. Nice. Very good. Okay. All right. So the think of osmolarity as like concentration of substance. That might be an easier way to think of this. So if the solution is hypotonic, the interior of the cell is relatively hypertonic, and therefore, we have a higher osmolarity inside the cell. The opposite will be true of a hypertonic solution, right? Oops. Good. I know there are a lot of different terms associated with this. I just know that in the nursing program, they like to use the term osmolarity a lot. But if you think of osmolarity as like the concentration of solute, how salty or thick it is, I think that will stick to you a little bit better. Very good. Good. So, just that anything with diffusion or osmosis in the title means that it's passive. Okay. So if, the question might have been, what is the, facilitated diffusion is active or passive? and then you're just going to put passive, because anything with diffusion or osmosis in the title, is passive. Facilitated diffusion, just think about what facilitation means. It means it just needs some help to diffuse. That's all. Nice. Very good. Oh, you can put hypotonic if you want, but I need to get rid of this question because it's actually not true. Actually, they can all. For some reason in the textbook, and I never really paid too much attention to it, they'd said that, it's not used. And it's like, it is used. And it, at one point I never really paid attention to it. Then I was like, I need to get rid of this. Because I was like, no. There are definitely applications of using a hypotonic IV solution, and that was confirmed by some notes I got from one of the nursing students. Okay, very good, you guys. I know this is kind of moving, but after the 2 chapters this week, we'll be able to kind of relax and prepare for our exam. So, okay, let me close these pages out. And there we go. All right, so we're going to go back to our notes and continue. Right, so the cell cycle. This is a series of changes a cell undergoes from, the time it forms until the time it divides. So, most cells are going to be capable of replicating themselves, and they need to, because you need to restore the population of cells that get worn out and taken out of circulation. So when it needs to make a carbon copy of itself, and these are called general body cells or somatic cells. These are not cells that are associated with reproduction, like oocytes and sperm. These are what's called somatic cells. So there are 3 stages, or major portions of it. We have interphase, which is a phase all by itself. Then we have mitosis, which has subphases to it called prophase, metaphase, anaphase, telophase, which functionally is to nuclear divide or divide the nucleus. So they're all going to be steps in how we break down the membrane of a nucleus, pull it apart, and then remembrane bound it. So we have interphase mitosis, and then cytokinesis is cytoplasmic division of the watery cytosol and all the organelles. And so how do we start with 1 cell and end up with 2 virtually identical cells? So we start with interphase. And interphase is the phase that the cell resides in the longest. 90% of its time, of a cell's time, is spent in interphase. And this is why. During interphase, we have to replicate everything. You have to replicate your chromosomes and DNA, or organelles, make extra proteins. You have to essentially make a carbon copy of everything. So when you split it in half, each cell takes with it enough organelles to exist, or at least get a really good start on existing on its own, and maybe make some, other proteins that it might need over time. So that takes a lot of time. You need to copy your DNA, you need to break the hydrogen bonds and begin in the middle of DNA, you need to make copies of everything, extra organelles, and that's why the majority of the cell's time is spent in interphase. Then we have, mitosis, and mitosis includes the remaining phases: prophase, metaphase, anaphase, and telophase. So now that we've replicated everything, we start in mitosis, which are all the steps and phases associated with dividing the nucleus. So what does that start with? Well, in prophase, the chromosomes, now that we've made a copy of them, because for us to copy the chromosomes, right, we need to be able to unzip the DNA through the center, which means it has to relax. And then when we make a copy of it, and now we have like 2 strands of DNA, they can all start condensing back down. And so the chromosomes are very relaxed, when you need to copy it. And when it's done, it'll condense back down again and become very dense and coiled. And that serves another function. They're easier to move around that way. So the chromosomes condense or coil. The nuclear membrane breaks down because we can't divide everything that's in the nucleus if we don't take apart the membrane. And those centrioles now have replicated, and they migrate to opposite ends of the cell, and they produce these Mitotic Spindles that are essentially like finger-like projections that are going to grab onto those chromosomes and pull them apart. So these 3 events are associated with prophase. Chromosomes coil and condense because they're easier to move around that way. Nuclear membrane disappears because the centrioles, mitotic spindles, can't grab the chromosomes if they're membrane bound. And that the centrioles migrate to the poles and start building these mitotic spindles. Okay, so then we enter into metaphase. And in metaphase, we line the chromosomes up right up in the middle in a linear orientation. So those mitotic spindles are going to move these chromosomes so that they're all lined up right through the center in one direction or another, but just in a linear orientation. During anaphase, now the mitotic spindles start to pull those chromosomes apart. So, I like to kind of use the analogy of at each of the centrioles, the mitotic spindles are like you're fishing and you cast your line out and let's say you get a fish and then you start reeling it in, now they start to reel the chromosomes toward those centrioles to the opposite ends of the cell. And you start to see them separate because the chromosomes in your slides and things generally will take up stain, and you can see, them being pulled apart. So chromosomes become separated from one another. They get pulled to opposite ends of the cell. And toward the end of anaphase, a cleavage furrow starts to begin. So what is a cleavage furrow? It's when the cell starts to pinch in the center and starts to essentially divide the cell into 2. So it starts to pinch in from one side and then the other. And then we have telophase. So now that we're pulling these apart, they're traveling to one end of the cell to the other. We're already starting to pinch in a little bit and now separate, the cell to form 2 cells now, is that a few more things need to occur. And 3 functions of telophase are the exact opposite of what you did in prophase. So now, we're going to remembrane bound the nucleus. We've pulled the chromosomes to opposite ends. Now we're going to put another membrane around them so we can keep them in a little ball. The chromosomes can now uncoil because we don't need to move them around anymore. They're just going to relax. The mitotic spindles all break apart, and they can kind of be reused for other functions, in the cell. But in any event, the 3 things that happened in prophase, the opposite is going to happen in telophase, because now this is the last step to dividing the nucleus. So, the mitotic spindles let go, we remembrane bound the nucleus, so now we have 2 nuclei going. The chromosomes start to uncoil, and cytokinesis is completed. So that the beginning of the cleavage furrow, or that cleavage furrow, is when cytokinesis begins, because you start to pinch into the center and start to find, a region right in the center that can be approximated, bringing the 2 plasma membranes together and essentially produce 2 cells. And so once these are all membrane bound, we finish dividing up the organelles and bring the plasma membrane together, and now we produce 2 daughter cells. So cytokinesis is the division of the cytoplasm and the organelles. It begins late anaphase with that cleavage furrow, and continues until now you have two cells. There's a ring of actin microfilaments that contract and pull the walls of that plasma membrane together. It doesn't happen just passively. That is an active process, until finally it pinches through the center and we produce two daughter cells. So, there are in lab what I call cell on stick models, so they're going to be little cells on stick, little models, right? That we use generally only, 5 of those models to represent interphase, prophase, metaphase, anaphase, and telophase. And so once they're put in sequence, a lot of this makes more sense. And if you are especially doing lab face-to-face here or at one of the campuses, you look at slides of this as well. All right, so there'll be slides generally set up on microscopes, hopefully, that one station will represent interphase maybe. And, what we do is we find an example of interphase and put it at the cell pointer. And we also put one of those cell on stick models there. So you can look through the microscope and go, okay, this is what it looks like on a specimen. And this is interphase on a cell on a stick model. And then you would go to maybe the next microscope, that would be a prophase, a metaphase, an anaphase, and telophase. So you can see this kind of progression that's occurring. So we can also see them here in these pictures starting up at about 1 o'clock, where we have a late interphase. And you can see in late interphase, you can see the plasma membrane still exists, or the nuclear membrane exists around the nucleus. And you can see some material in the nucleus, but most of your chromosomes are going to be all relaxed, because during interphase you're copying it all. You haven't condensed it yet. So you can often see some material in the nucleus, and you can see the nuclear membrane because we're containing it, right? Because interphase is not part of mitosis. Interphase is interphase, making carbon copies of everything. But then, we enter into prophase. So that's at about 2 o'clock there. So it goes prophase. Now the chromosomes are coiled and condensed. And so you see those dark purples in there where the nucleus, area is. Those are the condensed chromosomes. Right? And look, the nuclear membrane that was that blue kind of line, solid line around the nucleus, it's gone. Right? And we have centrioles that are migrating to opposite ends of the cell and building those mitotic spindles. You see, so the little image to the left-hand side of prophase, gives you like a little picture, a drawn picture that shows you, okay, there are the centrioles, they're migrating to the ends of the cell, and those purple lines between them represent mitotic spindles. Okay, so then we go to late prophase, don't worry about that, and go to the next one, C, which is metaphase. And that shows you both in the example of the actual cell going through the process to the bottom, and above that C, shows you how those chromosomes will line up in the middle. Right? And you can see those purpley kind of clusters of centrioles and their mitotic spindles to the right and left of it. Right? And the diagram above it is showing you, okay, those mitotic spindles are actually going to grab onto those chromosomes and line them up. And then we go to anaphase, which is about 6:30, 7 o'clock here. Right. And now look at the actual slide, the histological slide. You can see the dark purples being pulled apart. So what happens during anaphase? Chromosomes are being pulled apart. Right. Those mitotic spindles are pulling them to one end of the cell and the other, and demonstrated by the diagram above it. Right. Then we go into telophase. And during telophase, those chromosomes are now separated from each other. We need to pull them to the ends and remembrane bound them. And that's what's happening above it. So now the chromosomes, the dark blue, are pulled to one end to the other. You're starting to build a nuclear membrane around them. Right? The mitotic spindles have let go of them. The purple, little lines are no longer, holding on to those chromosomes. You start to develop that cleavage furrow. And that, well, actually, you started developing that in late anaphase, I will say, honestly. And that furrow becomes more exaggerated and separating, or closing down within the middle of that cell until finally you produce two daughter cells. And so we start with interphase, prophase, metaphase, anaphase, and telophase. And theoretically, especially if this is an active cell, is that once it's done with telophase and you have these two daughter cells, they go right into another interphase to then replicate themselves. So you start out like in here with one cell, you end with two daughter cells over at about 11 o'clock there. And then those two daughter cells will enter into interphase, prophase, metaphase, anaphase, telophase, and you'll end up with 4 cells, 8 cells, and so forth. Okay, so that's what you need to know for lecture and pretty much for lab. Cell aging, this is a genetic theory. Cessation of mitosis and cell aging are programmed into our genes, because our cells live for a long time. But, we often, as we get older, our cells or the cells that we're replicating sometimes have, they're a little faulty, or they're not, exactly, what we should be producing. And it has to do with what's called the telomeres. These are strings of nucleotides that protect the ends of a chromosome, like caps on shoestrings, and that's what I like to put that in there. So, most of, maybe not so much the shoestrings now, but sneakers, that would have the shoestrings would be made out of cotton fibers in there, and at the ends you had those little plastic tips, so as they were washed or beat up, that that the tips kept your shoestring from unraveling, because once that plastic became fractured or pulled off, the end of your shoestring would start to come apart and it would start to unravel. And you could always trim, maybe put some glue on it or something, but these telomeres are kind of synonymous with, or example of that would be the caps on shoestrings. And what happens is, as cells replicate themselves over and over, those telomeres, or those plastic tips, on the shoestrings, become shorter and shorter and shorter. Right? So every time a cell divides, the telomere shortens. So the telomeres may act like an hourglass on how many times the cell can divide. Telomerase is an enzyme that lengthens the telomere and helps maintain it for a longer period of time. These are found in germ cells of embryos, but absent in adult cells, except for cancer cells. And so that, explains a lot, because generally cancer cells are replicating without the normal inhibitions that are present for your normal body cells to replicate over and over again. So what I'm saying is that your normal body cells generally don't over-replicate. There are certain things in their environment making contact with cells like next to them that don't encourage them to keep replicating unnecessarily. They replicate kind of when needed. Cancer cells have a tendency to replicate over and over and over again, some rapidly more than others. Some cancers are faster growing than others. And that's because they're finding that they have more telomerase, and that their telomeres are intact for a longer period of time, perhaps. It's a genetic theory. Okay, so that takes us to another Kahoot, and I believe that's the end of it. Okay, and that's the end of the presentation. So let me stop that share and get into the new web page. There we go. Did I go into the wrong one? Hold on. Let me go to a different one. Here we go. Let's try some different music here. All righty. Nice, very good. Nice. Good. Good. All right. Let me get out of this. Close that page out. Okay. And before we move on, so I just want to go back and mention something real quick here, because when I start doing this and I'm lecturing like this, I didn't skip over it, but I didn't maybe emphasize this. So when we started, we were talking about this somatic division. Somatic means non-sex cells, so it's general body cells. And we're talking about interphase. IPMAT is kind of an easy way to remember the phases in sequence: interphase, prophase, metaphase, anaphase, telophase. Right. It's the PMAT that are phases of mitosis, just to be clear. Interphase is interphase. Right. That's the replication of all your organelles and your chromosomes and things, not the division of the nucleus. But prophase, metaphase, anaphase, and telophase, as we've now seen, all right, are all subphases, and parts of stages of dividing the nucleus and remembering binding it. And then you have cytokinesis, which is then division of everything else and bringing the plasma membranes together so that you produce two individual daughter cells now from the parent cell. Okay, and so that's the end of the PowerPoint. I'm going to stop sharing that, and let me just. Okay, so does anybody have any questions about what we covered?